Is that how that went? Okay. Okay. He'll tell you all about that. And Dennis worked for Upjohn, a legacy, big time company that has provided many benefits for all of us over the years. Worked for them for 30 years. So I give you Dennis Weber, and he's going to talk to us about how a drug comes to market.
Thank you, folks. Yeah, I was 19 years old and got married. Kids, Catholic and careless, did it. And consequently, I had to start to do something about it. So I went to the Upjohn company. I was a janitor, believe it or not. So I worked nights and went to school days at Western for a bachelor's degree in chemistry with math and physics minors. I graduated in 1958, and then because I had a chemistry major, then they put me in the laboratory at Upjohn as an assistant. So and then I continued going to school nights and weekends for a bachelor's degree in chemistry. I got that and six kids, and I didn't dare have any more kids because they'd have stopped my grant probably. Catholic and careless, see. So I spent 13 years at universities, came out with a PhD, worked at Syntex for about 3 years. Then I came back to dear old home at Upjohn in 1991, and I was spent 30 years total at Upjohn, researched drug metabolism, pharmacokinetics, and some of the clinical work also. And my talk today is about what the history of the pharmaceutical research situation in America. That's changed, by the way. The government has destroyed. By the way, I bought 18 copies of a book that I published. It's spread out here somewhere. Uh, Generics is the title. The destruction of the American pharmaceutical industry. And that's exactly what happened. I'll, we'll start with discovery, and I'll tell you a little bit of the history. Sorry, I got a big voice, but not big enough. Okay. Before 1900, the pharmaceutical industry mostly didn't exist. Pharmacists had to make their own pills, and they had what they called a pill tile, piece of glass or a flat. So they would put a little bit of the medicine on the tile and some kind of a binder. And then they would use their hand to roll it around to make a pill. And and after they did that, then they would sell it to the customer. The only problem was the binder that they put on the pill wouldn't dissolve in the water. So what went in with the mouth came out the back end the same as it went in. In other words, it didn't work. Didn't work. And there were some other things too. So consequently, the government decided that we'd better do something about it, and that's when the Food and Drug Administration was formed. And food, drugs, and cosmetics then were controlled, and they had to be safe, and they had to work by law. And that was the beginning of the modern pharmaceutical industry in the United States. Okay? Any questions yet about the beginning? Getting? Yeah. 1906 was the beginning of the Food and Drug Administration. And there's some more to that also. We'll get into it. So the pill tile that the pharmacist owned, by the way, there was a recipe book for all this. the United States Pharmacopoeia was a book. It was a book of recipes for how to make medicines, and that was the Bible for the pharmacists. Sorry about that. That doesn't work either. Okay, thank you. It's okay, folks. Keep me honest. Thank you, Chief. Amen. Okay. So consequently, in 1906, then the Food and Drug Administration was formed, and then the pharmaceutical industry began to be modern, and they didn't have any pull tiles. And we'll at the end of this thing, we'll talk a little bit about the manufacturing process of the pharmaceutical industry. They actually started off included cement mixers. I know you don't know why, but it's true. I'll tell you the story later. Okay, so the pills that the pharmacists made were too hard. They passed through the body, not absorbed. The United States Pharmacopoeia. There's a recipe book, the Merck Manual, Physician's Desk Reference, and the United States Pharmacopoeia, which was a book of recipes for how to make medicines. It, it was the Bible for the pharmacist. Okay, food, drug, and cosmetics. The Upjohn Company then was formed in 1886. The Upjohn family, several physicians, I think it was, started it. They're all gone, and I'll tell you the story of, of how all that happened. The Upjohn Company had more than 80,000 samples, chemical samples of substances that they were using to try to find some that had some biological activity that was useful. Okay? The process of doing all that was extremely expensive. Several hundred millions of dollars went into the research over 10 years. In those days, the product that you get out of the Walgreens now is not what's patented. It's the molecule that's in that box or in that package that's patented, not the product, just the molecule. So in the 1960s, they lied. It's the molecule and not the product. And there's more to that also. That's the beginning of the destruction of the American pharmaceutical industry. I have 18 copies of my book that I had published. That are on the tables here. You can buy new copies at the bookstore in Portage, $10, and they'll deliver it to your house if you want a personal copy. Okay, so then how do you discover a molecule that has some biological activity? The USP and the government says it's got to be two rodents, mice and a rat, and two mammals, cat and a dog. Those are the ones that were used to look for biological activity that might be useful. And that was the first step in the discovery process. Potential new drug tested for useful action in animals, two rodents, mouse and rat, and two mammals, cat and dog. Beagle dogs. Now, you probably already know that dogs love people. They love attention. Well, Upjohn had building 18 had 2 floors of animals. The first floor, not the first floor, but I think it was the fourth floor, was rats and mice, and the one above that was the beagle dogs. All you had to do was walk down the aisle, past the doors, and the dogs would know you're there. They'd bark like crazy. Come and see me, come and see me, come and see me. So if there was a molecule that was found to have some biological activity that looked that it was going might become a medicine, it was tested first for toxicity in rats and mice. And they would feed the material to the rats and mice in their food, then sacrifice the rat in the mouth. The animal was taken apart, and every tissue, right down to the eyeballs, were put on microscope slides, and experts with microscopes then were looking through hundreds, if not many hundreds, of, of microscope slides, looking to see if there was any damage done to the animal, the rat or the mouse, by the potential new drug. If they saw anything bad at all, that was the end. All research stopped on that molecule. They couldn't afford hundreds of millions of dollars on something that wasn't going to work. Okay, the desired bioactivity, as an example, one example is blood pressure. There was a machine that they designed. It's called a chymograph. It was a paper about this wide, smoked with a black with a pen scratcher on it, and then they would hook up the cat or the dog's heart to that, every heartbeat would make the scratch on the recorder, and they could tell then is it active or is it enough or is it not active. And this was the very simple, and these scientists then, that's all they did was test drugs, molecules, trying to find something that had some bioactivity of some sort. Every kind of test that you can believe, that you can imagine, was used. It wasn't only blood pressure, all kinds of things, and very expensive. Okay, if the desired bioactivity was found in a molecule, and the toxicity testing was acceptable, then it went on to the next stage, which was a development. And that's the brown envelopes that I have here. There's 3 stages listed: discovery, development, and then manufacture and marketing. And those are you can have them. Now, why was it important for the FDA to be formed? It was safety. That there were some antibiotics in the 1920s, uh, sulfonamides, antibiotics. It was a sulfa drug. It was an antibiotic, a powder. In fact, in the military would drip powder in an injury, in battle even. That's the medics. Well, it was the beginning, and science didn't understand everything that needed to be tested and looked at to be sure you have a useful, safe medicine. Well, what happened was that the sulfonamide antibiotic that was given to the civilians, the solubility, the solubility was such that it would not dissolve in the urine, so the body couldn't get rid of it. You got to get rid of the medicine, goes in, does the job, and leaves. But this stuff didn't leave. It destroyed the kidneys, and people died. That was the beginning of when the pharmaceutical industry realized, hey, it's not enough just to find out in some animals, we got to do a lot of deep, deep, deep science before we dare to have a safe and efficient medicine. And that was the beginning of the modern pharmaceutical industries. As you all know, Upjohn is gone. There's more to that story too. Upjohn, Park Davis, Squibb, most of Merck are all gone. The jobs are gone overseas, and our money is going overseas to Washington D.C. to re-elect some of those. I won't use the word. Okay, so now we have a molecule that has potential to become a product, and that's the beginning of the development phase. Can I get the first phase two, development? Upjohn, speaking about Upjohn, the, the big Portage had a lot of research pharmacists that designed the tablet and decided or made both capsules, tablets, maybe injections, because the development had to be done on all of these to find out which was best and which worked and which one didn't work. Animal tissue samples after a dose. In the microscope, can I show the curves? Okay, now there's two ways to dose a medicine. Oral, you swallow a tablet, or you get injected with a hypodermic needle. Well, if this is the intravenous dose, you inject it, and immediately it gets into the bloodstream. So the concentration along this line here is very high. Immediately, the body begins getting rid of it. So the concentration begins to fall. The mathematics of that curve is what I had to work with. The area that's underneath the curve represented the amount and the concentrations of the in the body of that medicine. When it gets way out on the bottom, eventually there's no more left. Okay. Now they had to decide what route to put whether it was an injection, tablet, capsule, or whatever it was. It had to work. This is the oral dose. Well, it starts off down here before they take the tablet. Let's say the tablet dissolves, I hope, and the medicine then begins to be absorbed into the bloodstream. And now the human liver, everything that we eat that gets absorbed into the bloodstream goes to the liver first, and the liver decides if this stuff is going to be safe enough and able to be kicked out eventually. So consequently, as this as the medicine passes from the intestinal tract into the bloodstream, the concentration of blood goes up. Eventually, though, then everything that's going to come in has gone in, and it's got the treat then. This line here is the concentration in the bloodstream that has to be higher than that in order to cure the disease. The top line is the toxic level that you don't want to get up to. So consequently, in this situation between here and there is where the concentrations in the animal bloodstream had to be in animals and humans. How did they determine that? By the toxicity result in the animal. Is what they did. Highly trained but uh, scientists that could look in a microscope slide and tell if that was good tissue or damaged tissue. They knew what they were doing. Okay, now, if the usually the molecule that they saw some activity with isn't useful enough to put into a dosage. Can I get salicylic acid? This thing, this one. Okay, salicylic acid. Now, aspirin is acetyl salicylic acid. What they do, they react this with acetic acid, vinegar, so to speak. You get that here. Now you have aspirin, and then that then becomes the chemistry that has to be in production in order to make the dose that's going to actually go into the humans. The the sulfur drugs, sulfur meaning sulfur. I mentioned already that they made a mistake and the solubility was not enough. The body couldn't get rid of it, and it destroyed the kidneys, and the patient died. So now they have triple sulfurs. What they do, they take three different sulfur molecules, all of which will dissolve in the urine and dissolve in the body and be eliminated. Okay, so the triple sulfur antibiotics now are sold and they work because the body is able to get rid of all three, and none of the three stay in the body. They all get rid. So you want the medicine to get in, do the job, and get out. That's part of it. There's another one. I had a personal work several years on something called U67590A. That's an Upjohn code for a molecule. Now, when we have a brain bleed, blood contains iron. Iron exists in two forms, the ferrous and the ferric. Ferrous is +2 charge, and the ferric is +3 charge. That sets up a process of continuing damage to the brain. That's why when somebody that has a brain bleed, there's a continuous damage done for weeks, many weeks, even after the damage occurs first. It's a continuous damage. So consequently, the U67598 methylprednisolone was the name, stops the process with the iron, and it's still in the market, and it's a very, very important molecule, folks. U67598. But they have to determine the dose, don't they? And they did. And the volunteers, and there were volunteers at Bronson Hospital. My son was one of them. Would take a dose, very small, very, very small dose, and they would raise the dose just a little bit at a time, tiny bits amounts. And the very first time that there was anything that they saw that was not correct, it was the end. Never again would that molecule be given to a human. It had to be, and this was in the volunteers. Now, this is not the clinical studies, that's something else. This was safety. Okay, so it's called solid The molecule was called solumedrol. Medrol is a steroid. Oh, and by the way, with steroids, I'm kind of special with this, and I'll tell you why. Steroids were very important. Science knew that there were steroids in the body. It was part of a very, very important part of the body chemistry. And John Moll, the pharmaceutical company, wanted steroids to test, trying to make a medicine using steroid molecules. But they couldn't find the steroids. Well, it turns out that Borges Hospital maternity ward, pregnant women's urine has a lot of steroid in it. So Upjohn was buying the pee of the pregnant women at Borges Hospital up on the top floor, including my mother's pee. So I'm the piss paid kid. And that's the aspect. She got greedy, though. She tried drinking too much water, so they fired her. And then what they did, they went to horses. Over on G Avenue, pregnant horses had the steroid, so they were raising horses to get the steroid. And then there was a lot of research being done on the Upjohn source of steroid. But then in Mexico, there's a tuber, like a potato, but this big around, full of raw steroids. Nothing medically useful, but it was a steroid. Holy smokes! Upjohn bought truckloads of that stuff, rounded it all up, and extracted the steroid molecule out of it, and that was the beginning of steroid analysis and the development at the Upjohn Company and every other pharmaceutical company. It was Syntex, a Mexican pharmaceutical company that was selling these tubers with the Roundup, the steroid in it. And that went on. How did they know it was going to be work? Well, there was a scientist at Upjohn. So what he did, he took the raw steroid that they got from Syntex and fed it to some germs in the diet. And sure enough, the germs ate it, and you know what they did? They spit out a steroid that was different and could be made into a medicine. And that was the beginning of Medrol, Solumedrol, that became a very important medicine for people who had brain bleeds. And it's still on the market, folks. It's still there. But U67-598 worked wonderfully in men, but the dose for women was different. That meant they had to start all over again. Upjohn canned the whole thing. It was done. U67-598 never got to market because they had to do it all over again because the dose in the women was different than the dose in men, and the research would have to be repeated. That's just an idea and a suggestion of how much work had to be done to put anything on the market as a medicine. Hundreds of millions of dollars were done. And in those days, a 15 years was the limit for a patented molecule for the patent. It took 10 years to take that medicine and try to make a drug out of it. So there was only 5 years left before someone else could start using that molecule if they knew what it was. And that was the kind of information that was not put out. Okay, now that's the part of this discovery. We now have a molecule that has useful biological activity And now the next phase, phase 2 on your sheets, is the development phase. Okay? Can you hear me? Okay. Well, first of all, then, there's what a research pharmacist had to design a tablet, a capsule, injection. Uh, how did they determine this? From animals and dogs. The beagle dog, by the way, they they use beagles for the testing. If you walk down the aisle, the corridor, all the dogs are over here. They bark like crazy. Come in and see me. Come in and see me. Well, we had a table about this size, this high, where the dog would we put the dog up here and to take a blood sample out of the vein here. The dog had been dosed with the new medicine. Well, the dog was so anxious to be with humans and to have some attention. This is the honest truth. The dog would jump up on the table and do this. Do me what thou wilt. Then they take the blood sample, and then the blood sample would come to me in the laboratory, an analytical chemistry procedure that I developed. And then I had to measure the amount of drug in that small blood sample. And they took multiple samples as a function of time so they could see the rise and the fall in the concentrations like we saw before here. Yeah, that was the first stage in the development. Toxicity then had to be measured. And eventually, if everything looked okay, then the pharmaceutical company would collect all of the data in all of the research notebooks that were in the lab that the scientists wrote what was going on in the book. All those books went to the Food and Drug Administration, the FDA. If the pharmaceutical company decided that it was good enough, it had to be approved by the Food and Drug Administration. And it took several years to go through all of that truckfuls of data. And if the FDA decided that it was going to be okay, then they would get a permission to go to the human clinical studies. Okay, and then the Bronson Hospital had a clinical department also. It was called an INDA, Investigational New Drug Program. These were volunteers that volunteered to take the test drug, and how much did they? How did they know how much to give the volunteers? Well, way back years before when they were giving it to rats, they kept raising the dose in the rat until the guys with a microscope said, "Whoops, something bad has happened." That concentration that caused a problem, a toxic problem in the rat, was the beginning of the information of how much to put in the human. They would divide that dose by thousands of times down to micrograms. Very, very, very small amounts would go into the human volunteers first, and then they would go up, 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 slow, slow, slow. And oh, by the way, they locked the doors to the bathrooms. By the way, why? Because they had to collect all the urine to find out how much drug was coming out in the urine. You, you collected the blood and the pee. Okay, and that again was part of my job. That was the clinical work that I helped with. I had to develop chemical analytical procedures to measure tiny, tiny amounts of drug in blood and in urine. and then plot and it's called pharmacokinetics, the rates of increase and decrease of the concentration of the drug in the bloodstream of the volunteer. Now, if everything looked fine, but if there was anything in that human volunteer that was not normal, that was the end. They stopped. And never again would any human get a dose of that size. If that dose was less than the needed dose in humans, they were done. They had to start all over again with 10 years and start all over again and start hunting for something else. That was the need. They had to find what the toxic level of that medicine is in the human body. And the volunteers, and it didn't make a difference what it was. I mean, anything that was not normal, it was done. And they dropped it all. The solumedrol one with to dissolve it in the brain, it works beautifully in men. It went all the way to the not the market. It was still in the clinical studies, but the dose in women was different. So they had to repeat everything in the women's positive situation. So they dropped that too. U-67-598 never made it to market after 10, 12, 15 years of extremely hundreds of millions of dollars of expense. So then what happens? Well, all the data that's been collected on toxicity, biology, human results, and so forth were sent to the Food and Drug Administration. And I mean, there were truckloads of research books, data that the FDA had to look at. It took them years, 2 or 3 years or more, to try to make a decision whether or not the company was allowed to make something and put it on the market. And that's what the pharmaceutical company was waiting for: permission to market the product that they had found. And it wasn't always true that you were going to be able to market. The FDA was tough, and they had to be tough. It killed a whole lot of guys back in the 20s with the antibiotic problem, and that was not going to happen again. Okay, and then the dosage regimen, oral those tablets, they had to look at the toxicity level. What is the toxic level of the drug in the blood that you must not get above, and then the level that you have to get above to be useful? The mathematics of that curve had to be done. It's the area under that curve that was the total dose that was in the body. Mathematics, physics, and chemistry, all three of them, and a hell of a lot of biology was necessary. And that kind of studies comes expensive. And that's why it was hundreds of millions of dollars just to get to the point where the FDA was going to decide whether you could or could not market that product. And sometimes they said no. And they started all over again. Most of the companies, Upjohn's included, were started by physicians. The physicians knew the problems that they had to be solved in order for their patients to be treated well. It was the Upjohn family, bunch of physicians. Park Davis were physicians. Upjohn, Park Davis Squibb was a family of physicians. Merck, again, was that. The physicians that are smart enough because they knew what needed to be done, and then along came the government. In 1960, there was a senator, Estes Kefauver was his name, and before 1960, all of the over-the-counter medicines that you see in Walgreens, you see hundreds and hundreds of different kinds of stuff. It had to be safe, but it didn't have to work. And it didn't work. So Estes Kefauver, Senator Estes Kefauver, said, we want everything to work. Sure enough, he put together enough screwballs to pass a law that everything had to work. Well, the over-the-counter stuff, he spent the next 10 years, from 1960 to 1970 at least, testing all the over-the-counter drugs to find out which ones worked and which ones didn't. The ones that did not work were told, okay, get off the market or fix this thing. And they went out of business. A lot, Carter's little liver pills, and the pig stuff, gone. And then Kennedy got assassinated in 63. Johnson, typical politician, became free just for the asking, no cost. All of the information and stuff that the pharmaceutical companies are medicines on the market, and he did it. Now that meant, and remember, it's the molecule that's patented. When the patent runs out, that's okay, and some other company can take that molecule and make a new product. But they have to do all of the research, toxicity, everything else again, and the over-the-counter people didn't want to do it. It was too expensive. So they wanted permission to not only get the molecule, they also wanted to be told how to make the medicine. In other words, all of the information and work for 10 years at least, the manufacturing processes even had to be known. So Johnson said, yeah. So Johnson gave permission for those companies that did not, whose products did not work, everything that they asked for to make the product that was already on the market. That meant that the pharmaceutical companies that were doing all the research could not recover the cost of hundreds of millions of dollars of the research. They had to go out of business. Upjohn stayed on into the early 70s, but eventually they went broke. They could not make any money because they could not recover the years of research to make a safe and useful medicine. Excuse me. And that's why we don't have Upjohn, Park Davis, Squibb, most of Merck. With the insulin, diabetes, you don't cure diabetes, you just control it. Eli Lilly's still in business because and and then those companies overseas did not have the sales uh, information that they needed, so they went to Merck, Merck couldn't do it anymore. They were going broke. So, but all the pharmaceutical companies had a information source. They had employees, pharmacists that would have an appointment with a physician, and it was the job of the visiting pharmacist to give the physician all the information needed to convince the physician that this was going to be a useful product. And then it was the job of the physician to write the prescription, and that was the point when the pharmaceutical company knew that they had something that was useful. And it's still that way today. However, the people, Upjohn required all of their. By the way, those pharmacists were called detail men. They would give the details of a usefulness of a new medicine to the physician, and to convince the physician that this was going to be a useful and safe medicine. And then it was the physician's job then to decide whether to write a prescription or not. And the prescription then was the key event that the pharmaceutical companies were always looking for it to happen. And it did, as long as they could recover the hundreds of millions of dollars of research costs and therefore stay in business. When that happened, it all ended, folks. Pfizer is not a pharmaceutical company. There's no research there. They make medicines, but it's not a pharmaceutical. It's a manufacturing company, is what it is. Oh yeah, and by the way, I told you the story about the pill tile where the pharmacist would make pills. Well, they had to change that, so now they have a cement mixer style. Yeah, small, just pharmaceutical quality cement mixers, stainless steel. And what would happen was the small bit of the medicine molecules were they would get thousands of these little pellets, not the dose, just a pellet, and they would put it into the research quality concrete mixer, and it would stir it, and then they would spray with water soluble stuff on top of that, and it would get distributed, and then when everything was done, then they would take all of the pills that they were in the research in the manufacturing process and take it to the bottling people, that they would put them into bottles and label them. And there was always a little piece of cotton in the top, and they did that so that so that the pills didn't rattle around and get broken. So they were uh, the cotton was there for a reason. It worked. All kinds of jobs. Uh, Kalamazoo people, Kalamazoo families often had a picture of Dr. Upjohn on their wall. My hero, so to speak. Okay, and uh, and there's one more phase, phase 4. After a medicine is on the market, there's a continuing, never-ending checking of how that medicine is working in the population. It's called phase 4. In other words, after it gets to the market, they're still looking to look for its safety and utility. Any questions? I know you got a lot of them, but you don't know how to ask them. It's okay. I've got 2 books up here, the Merck Manual. By the way, you can buy a copy of the Merck Manual at the Barnes and Noble bookstore. You have to order it. And I also have a copy of the Physician's Desk Record (PDR). Years ago, it was a book that the physicians used to look up the properties of the possible medicine. Now it's on the internet, so they don't need the book. That's how I got a copy. Any questions?